Welcome to Botany class, Chapter 4, Photosynthesis in Higher Plants. Today's topic, the electron transport. In photosystem 2, the reaction center chlorophyll A absorbs 680 nm wavelength of red light causing electrons to become excited and jump into an orbit further from the atomic nucleus. These electrons are picked up by an electron acceptor which passes them to an electron transport system consisting of cytochromes. This movement of electrons from phaeophyton to P700 is a downhill. In terms of an oxidation reduction or redox potential scale, the electrons are not used up as they pass through the electron transport chain, but are passed on the pigments of photosystem 1. Simultaneously, electrons in the reaction center of PS1 are also excited when they receive red light of wavelength 700 nm and are transferred to another acceptor molecule that has a greater redox potential. These electrons then are moved downhill again, this time to a molecule of energy rich NADP+. The addition of these electrons reduces NADP+ to NADPH plus H+. This whole scheme of transfer of electrons starting from the PS2 uphill to the acceptor down the electron transport chain to PS1. Excitation of electrons transferred to another acceptor and finally downhill to NADP plus causing it to be reduced to NADPH plus H plus is called the Z scheme due to its characteristic shape. This shape is formed when all the carriers are placed in the sequence on a redox potential scale. The electrons that were moved from photosystem 2 must be replaced. This is achieved by electrons available due to splitting of water. The splitting of water is associated with the PS2. Water is split into photons. These oxygen and electrons. This creates oxygen, one of the net products of photosynthesis. The electrons needed to replace those removed from photosystem 1 are provided by photosystem 2. 2H2O gives rise to 4H plus O2 and 4 electrons. Here, water splitting complex, oxygen evolving complex, it is called as OEC, is associated with the PS2, which itself the physically located on inner side of the membrane of thylakoid. So you should remember this bit. They may ask indirectly in the NEET examination part. Next topic, cyclic and non-cyclic photophosphorylation. Here, capability of extracting of living organisms energy from oxidizable substances and storing this in the form of a bond energy. Special substances like ATP carry this energy in their chemical bonds. The process through which ATP is synthesized by cells in mitochondria and chloroplasts is named photophosphorylation. Photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of light. When the two photosystems work in a series, first PS2 and then PS1, a process called non-cyclic photophosphorylation occurs. The two photosystems are connected through an electron transport chain. In Z scheme, both ATP and NADPH plus H plus are synthesized by this kind of electron flow. Only PS1 is functional. The electron is circulated within the photosystem and the phosphorylation occur due to the cyclic flow of electrons. Here, a possible location where is this cloud be happening in the stroma lamellae, while the membrane of lamellae of the grana have both PS1 and PS2. The stroma lamellae membranes lack PS2 as well as NADP reductase enzyme. The excited electron does not pass on to NADP but is cycled back to PS1 complex through the electron transport chain. The cyclic flow hence results only in the synthesis of ATP but not NADP H+. Cyclic photophosphorylation also occurs when only light of wavelength beyond 680 nm are available. In green plants, cyclic photophosphorylation is an additional source of ATP required for chloroplast activities over above the is required in the Calvin cycle. ATP is synthesized in the chloroplast. The chemiosmotic hypothesis has been put forward to explain mechanism. Like in respiration, in photosynthesis too, ATP synthesis is linked to development of a proton gradient across a membrane. This time, these are membranes of the thylakoid. There is one difference through. Here, the proton accumulation is towards the inside of the membrane. In the lumen, while in respiration, protons accumulate in the intermembrane space of the mitochondria when electrons move through the ETS. What causes the proton gradient across the membrane? We will discuss right now. Electron and their transport to determine the steps that cause a proton gradient to develop. First step, splitting of the water molecule takes place on the inner side of the membrane. The protons of hydrogen ions that are produced by the splitting of water accumulate within the lumen of the thylakoids. Second, electrons move through the transport chain. Protons are transported across the membrane. This happens because the primary acceptor electron, which is located towards the outer side of the membrane, transfer its electron not to an electron carrier but H carrier. Hence, it removes a proton from the stroma while transporting an electron. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into the inner side or the lumen side of the membrane. Proton gradient across the membrane increases due to quinine cycle flow of electron between P and Q and cytochrome B. Third step, the NADP reductase enzyme is located on the stroma side of the membrane along with electrons that comes from acceptor electrons of PS1. Photons are necessary for the reduction of NADP plus to NADPH plus. These protons are also removed from the stroma. Hence, within the chloroplast, protons in the stroma decreases in number, while in the lumen there is an accumulation of protons. This creates a proton gradient across the thylakoid membrane as well as a measurable decrease in a pH in the lumen. So, this gradient is important because it is the breakdown of gradient that leads to release of energy. The gradient is broken down due to the movement of protons across the membrane to the stroma through the transmembrane channel of the F0 of the ATPase. The ATPase consists of two parts. One is called as F0 is embedded and the another is the membrane forms channel that carries out facilitated diffusion of protons across the membrane. Other proton is called F1 and produces on the outer surface of the thylakoid membrane on the side of the faces the stroma. The breakdown of the gradient provide enough energy to cause conformational change in the F1 particle of the ATPase, which makes the enzyme synthesis several molecules of energy packed ATP. Chemoosmosis require a membrane, a proton pump, a proton gradient and ATPase. Energy used to pump proton across the membrane to create a gradient or high concentration of a proton within the thylakoid lumen. ATPase as a channel that allows diffusion of protons back across the membrane. The release enough energy to activate ATPase enzyme that catalyze the formation of ATP. Along with the NADPH produced by the movement of electrons, the ATP is used immediately in the biosynthetic reaction taking place in the stroma for fixing of CO2 for the synthesis of sugars. For more videos, download Camel4U app. You can get another information.